as Lakshmi said, my interest is in random receptor calcium release channels, primary efflux pathway for calcium to initiate contraction of the heart and skeletal muscle. So I'm going to start with just a brief introduction of random receptors and calcium release. Then I'll talk about some of the methods we use in our lab. Then I'll talk briefly about 3 different projects we've got going. This S-adenosyl L-methionine, calmodulin, and some functional studies of the channel from aged muscle. And along the way, I'll try to mention some future directions of where we plan to go. So I think as most of you know, cells maintain intracellular calcium concentrations at quite low levels, somewhere around 50 to 100 nanomolar. That's in spite of the fact that extracellular calcium is in the millimolar range, and most cells have a calcium storage organelle, usually the endoplasmic reticulum if you're not an excitable cell, if you're not a muscle cell, or the sarcoplasmic reticulum if you're muscle. And in the sarcoplasmic reticulum, calcium concentrations are around the millimolar level. So there's a huge driving force for calcium entry into the cytoplasm, which is good if you want to use calcium as a signaling molecule, because you can just have a relatively small change in the permeability of the membrane to calcium, and you have a significant amount of calcium entry. The downside is that calcium is cytotoxic. So if calcium is elevated too high or for too long, you can kill off your cells. So cells maintain this balance through the intricate interplay of numerous calcium channels and pumps and exchangers. So there are ATP-dependent calcium pumps on the surface membrane and in the calcium storage organelle. So they use ATP hydrolysis to pump calcium either out of the cell or into the storage organelle. There are channels in the surface membrane. These can be voltage regulated or ligand gated that allow calcium influx. There are calcium release channels in the ER or SR. The inositol trisphosphate receptor is one, and its cousin, the random receptor, which we'll talk about. Then there are calcium exchangers, things such as the sodium-calcium exchange, which use the concentration gradient, electrochemical gradient for sodium entry to drive calcium out of the cell. And this pump is actually reversible, depending on the concentration gradient in your membrane potential. So it's the interplay of all these channels and pumps that regulate calcium and keep cytoplasmic calcium at low levels. So calcium signaling is used for a huge variety of purposes. It's one of the first signals that occur upon fertilization of the cell, and one of the last signals that occurs prior to cell death. And in between, you use calcium for things as memory, gene regulation, regulation of metabolism, and my primary interest, muscle contraction. So focusing more on skeletal muscle contraction as a way of summary, this is kind of a cartoon of how your muscles get activated. So this is the muscle surface membrane, and somewhere down here at the end is the neuromuscular junction. You generate an action potential, which spreads over the surface membrane, and then down into invaginations of the surface membrane known as transverse tubules. Down here, depolarization of the transverse tubule triggers calcium release from the sarcoplasmic reticulum via the random receptor. So we'll talk a lot about this in a moment. So calcium is released, cytoplasmic calcium goes up, it binds to a calcium binding protein, troponin, causes the conformational change of the troponin trypomyosin complex, which lines the actin filament. That will expose binding sites on actin to allow the myosin heads to reach up and undergo the Qualls-Bridge cycle, causing the actin and myosin filaments to slide past each other and muscle to contract. This cycle continues as long as there's ATP and calcium. To relax the muscle, calcium is pumped back into the sarcoplasmic reticulum via the sarcoplasmic endoplasmic calcium ATPase, or SERCA. Cytoplasmic calcium falls and the muscle relaxes. So if you take a little closer look at a skeletal muscle fiber, here's the muscle fiber, kind of a cross section with some of the membrane peeled away. You see the surface membrane with these holes every so often. So these are actually the entries, the mouth of the transverse tubules. Transverse tubules are invaginations of the surface membrane, so they're continuous with the surface membrane. And they're shown here in yellow. They're actually a very, very extensive network in your muscle cells. In fact, the surface area of the transverse tubules is about the same as the area of the surface membrane. So it's a huge, huge membrane network. So as they enter the muscle fiber, they're abutted on each side by sarcoplasmic reticulum. So that's kind of this greenish gray color here. The sarcoplasmic reticulum, as you can see, kind of has two different domains. This is fat section, which is also known as the junctional sarcoplasmic reticulum or the terminal cisternae. Then this longitudinal network is the longitudinal SR. So it's this region of the triad where you have the transverse tubule abutted on each side with junctional sarcoplasmic reticulum where calcium release occurs. If you look at an EM, you can actually see the transverse tubule abutted on each side with sarcoplasmic reticulum. If you look closely, you see these electron-dense structures here. These are actually the ryanodine receptor calcium release channels. One point I should make is that in most experiments I'll talk about, we isolate sarcoplasmic reticulum through a number of differential centrifugation steps, and we can separate this longitudinal SR from the junctional SR. The junctional SR is also sometimes known as heavy sarcoplasmic reticulum because it's more dense on the sucrose gradient. The longitudinal is light SR, so we usually use the heavy SR in most of our experiments. So excitation contraction coupling, regardless of whether it's cardiac or skeletal muscle, and I'm not going to talk about smooth muscle, that's kind of a special case. It's really the story of two calcium channels. The L-type calcium channel that's in the transverse tubule membrane and the ryanodine receptor, which is in the sarcoplasmic reticulum. So the L-type channel is also known as the dihydropyridine receptor, or sometimes called the voltage sensor, depending on what function you're studying. It's known as the dihydropyridine receptor because it binds a class of drugs known as dihydropyridines. These are clinically relevant as they're calcium channel blockers and used for regulation of blood pressure and to modify the strength of the heartbeat. It's a heteromultimeric protein. There are five different subunits, and it's the origin of what is known as the transverse tubular charge movement in skeletal muscle, and also the L-type calcium current. The ryanodine receptor is also known as a junctional foot protein because in those EM images back in the 60s and 70s, people for some reason thought they looked kind of like feet. Sometimes also known as the calcium release channel. It's a huge channel. It's the largest ion channel known. It's a tetramer, homotetramer. The molecular weight of the whole thing is around 2 million Daltons. So just to compare skeletal muscle excitation contraction coupling with cardiac muscle excitation contraction coupling. They both use sarcoplasmic reticulum and T tubules. They both use L-type calcium channels and ryanodine receptors. They're just different isoforms and different coordination between the molecules. So in skeletal muscle, the dihydropyridine receptors, or the L-type calcium channels in the transverse tubule, actually form these tetrads. So there are 4 of these calcium channels clustered together, and they physically interact with a ryanodine receptor that's in the sarcoplasmic reticulum. I mentioned they're the source of this transverse tubular charge movement. So when the transverse tubule polarizes, there are charged residues in the L-type calcium channel that undergo a voltage-driven conformational movement across the electrical field of the membrane. Because you have charges moving across the membrane, you can measure that electrophysiologically as a sort of a capacitive charge movement. But that voltage-driven conformational change in the L channel is communicated to the ryanodine receptor through a mechanism that's not really clear. We're still trying to figure out the exact interaction between the two and how the movement of one channel opens the other. But that opens the ryanodine receptor. Calcium flows out. In skeletal muscle, these dihydropyridines, the L-type calcium channel, the pore-forming subunit is CAV 1.1, and the ryanodine receptor is isoform 1. One other thing I should mention is that in skeletal muscle, not all renin receptors are coupled to these L-type calcium channels. In very fast contracting muscle, there's a toadfish swim bladder, which was kind of the classic muscle to study the architecture of the triad. It was noted that every other renin receptor was coupled to one of these triads. So in this particular muscle, about half the renin receptors were uncoupled. In slower contracting muscle, the ratio seems to be even lower. There are even fewer coupled channels. So this charge movement or this conformational change in the L channel opens the coupled channels. The question then became, well, how are these uncoupled channels opened? There are two possibilities. One is if you look at the architecture of the triad or the face of this SR, you see kind of a checkerboard pattern of renin receptors. So the channels actually contact each other at the corners. They're kind of mushroom shaped or square shaped, so at the corners they actually contact each other. So it's possible there's an allosteric coupling between coupled and uncoupled channels, which will open the whole cluster of channels. The other hypothesis is that calcium released from the coupled renin receptors actually diffuses over and binds to the neighboring channel and causes the channel to open via a mechanism called calcium-induced calcium release. Now there's a basis for that calcium-induced calcium release in cardiac muscle. So here, again, you have transverse tubules and you have renin receptors, but the L-type calcium channel doesn't form these clusters of tetrads. They're found as individual channels, and also they're not physically coupled to renin receptors. So when the T tubule depolarizes in the heart, you have the charge movement, but this charge movement is involved in opening the conduction pathway of the L channel. A small amount of calcium flows in, binds to the renin receptor, and causes the renin receptor to open. In cardiac muscle, you have the isoform 2 of the renin receptor, and the L-type channel here is the pore-forming subunit is CIV 1.2. So here you have calcium-induced calcium release. Here it's mostly mechanical coupling, or at least the immediate mechanism. This is the renin receptor images reconstructed from cryo-EM. This is what the renin receptor would look like if you were in the transverse tubule looking at the channel. Here's the channel if you're in the T tubule, in the SR looking out, and the side view. So this TA is a transmembrane assembly. So this part spans the sarcoplasmic reticulum, and all of this sticks out into the cytoplasm. So this, about 3/5 of the channel, is what you see here in the EM. As I mentioned, it's the largest known channel. It's a tetramer. Each monomer is about 5,000 amino acids. This thing's so big it's unlikely to be crystallized anytime soon. So the best images we have are from EM. There are 3 isoforms, RYR1, RYR2, and RYR3. RYR1 is a predominant form in skeletal muscle. So if you isolate renin receptors from skeletal muscle, you get greater than 95, maybe about 99% RYR1, and you get a tiny amount of RYR3. If you isolate channels from the heart, you get almost all RYR2. Unfortunately, there's no tissue that you can isolate RYR3 from. RYR3 has a wide cellular distribution, and it's usually found in low abundance and often with some of the other isoforms. So RYR1 and RYR2 have been fairly well studied, RYR3 less so. Should also mention that all these channels are found in different cell types, but like I said, they're usually found with the other isoforms. It's only skeletal muscle and cardiac muscle that have predominantly one form or the other. The amino acid sequence is about 66% identity between the 3 isoforms, and the divergence falls mostly in three different regions, which seem to compose some of the regulatory sites for these channels. Functionally, these channels have a very high conductance, 100 picosiemens for calcium. Compare that to the L channel, which is about 20 picosiemens. But this high conductance comes at a cost of a fairly low selectivity. So these things conduct potassium, pretty much any cation, which is an advantage because in some of our single channel studies, where we use potassium, which gives us a very high conductance, a large current, it gives us very, very good signal to noise ratio. In vitro, in vitro, these channels can be activated through calcium. So regardless of the mechanism the channel activity that you study, whether it's ryanodine binding, which is a measure of open channels, single channel recording electrophysiology, calcium release flux, what have you, it exhibits a biphasic calcium dependence on calcium. So ryanodine receptor type 1 has this biphasic calcium dependence, as does RYR2. RYR2 has a higher activation by calcium. So it's activated to a greater extent solely by calcium, and there's a broader calcium dependence. This is a bit exaggerated, but it gets the point across. This kind of makes sense since this channel is the one that's activated by calcium in the heart, whereas RYR1 is activated mainly through this physical mechanism. So this biphasic calcium dependence arises from at least two calcium binding sites. There's one site that has a high affinity that when it binds calcium will activate the channel. So it causes the activation. There's another site that has a lower affinity for calcium that when it binds calcium, the channel closes. That causes the closing on this side. These sites also bind other divalent cations such as magnesium, which is physiologically relevant. Then there's an activation site on the luminal side. It's been known for quite a while that if the SR of your heart or skeletal muscle gets overloaded, that you can get spontaneous calcium release. This has particular consequences for cardiac muscle and may lead to some cardiac arrhythmias, which I'll mention in a minute. It's not clear whether this luminal calcium binding site is actually on the channel, or it may be mediated through calcium binding proteins that bind to the channel. So these channels form these large complexes with lots of accessory proteins. This is just some of them. So here's your RYR2. It binds a number of kinases such as PKA and CAM kinase. It's phosphorylated by those. There are also protein phosphatases, PP1 and PP2A, bind through their anchoring proteins. Phosphodiesterases are located right there on the channel. So are some calcium binding proteins such as sorsin and calmodulin, which I'll talk about a little bit more later. Homer is an accessory protein that seems to be involved in possibly anchoring the channel in place. FKBP is FK506 binding protein. It seems to be involved in maintaining coordination of the subunits so that all the subunits move in a coordinated fashion to open the channel. In the skeletal muscle channel, if you remove this, you get, which seems to be some uncoordinated gating, so you have channels that gate to different conductance levels. There are proteins that bind to the ryanodine receptor that span the SR membrane and bind to proteins in the SR lumen, such as calsequestrin and this histidine-rich calcium binding protein. So both of these have low affinity but a high capacity for calcium binding. They keep free calcium relatively low in the SR, but act as sponges, so they keep the total calcium relatively high. As calcium is released to the ryanodine receptor, calcium comes off these calcium binding proteins and maintains free calcium in the SR. These may also be the calcium sensors, which are linked to that activation of the channel by calcium overload. Also, mutations in calsequestrin lead to a cardiac disorder, which I'll mention in a minute. Some of the regulators of the channel, there's a huge number of them. Probably not a surprise because the channel's so big. But some of the endogenous ones are adenine nucleotides and calmodulin, which I'll talk about. Calcium and magnesium I mentioned. Hydrogen. During skeletal muscle work or in cardiac ischemia, the pH drops inside the muscle. That can affect the calcium release channel, as does inorganic phosphate. You hydrolyze a lot of ATP and create a phosphate. Phosphate goes up in the cell. That will have a significant impact on the channel. The dihydropyridine and the FKBP I mentioned. And then the channel is also subject to reactive oxygen species or reactive nitrogen species modification. Exogenous regulators. Caffeine's been known for a long time to cause muscle contraction. If you take an isolated muscle, dump a lot of caffeine on it, the muscle will contract. It acts by binding to the ryanodine receptor and causing the channel to open, releasing calcium. I once did a calculation. It takes millimolar levels of caffeine to activate the channel. To get enough from coffee, I calculated once, it's something like 50 to 100 cups of coffee within a very short time period. So the amount of caffeine that you would drink would probably kill you by other mechanisms before it caused your release channels to open. Well, Ryanodine is a regulator of the channel. It gives the channel its name. Ruthenium red, volatile anesthetics, these depolarizing muscle relaxants I'll talk about in relation to a muscle disease. Oxidizing, reducing agents, no surprise if reactive oxygen species can regulate it. Anesthetics, local anesthetics, tetracaine, close the channel. 4-chloro-M-cresol, I'll mention in a minute in regard to some of the studies we've done. Dantrolene is interesting. This is actually an antidote for a muscle disease, and ICAR is also a potential therapeutic agent for malignant hyperthermia, which I'll mention now. So there are a couple diseases linked to the renin receptor. In RY1, there are mutations that cause something called malignant hyperthermia. Another disease is central core disease. And there are a few other minor ones. RY2 has, I guess it's been in the last 10 years, they've identified mutations in the channel that cause two diseases that lead to arrhythmias: catecholaminergic polymorphic ventricular tachycardia and arrhythmogenic right ventricular dysplasia. I'll describe those a little bit more in a minute. And in RY3, there haven't been any diseases linked to it yet. It could be that mutations in RY3 are lethal. Could be they don't cause a disease. Could be that it's just not studied as well as RY1 and 2, and there just haven't been the diseases linked to it yet. So, malignant hyperthermia is the skeletal muscle disease. It's an adverse reaction to mostly to volatile anesthetics. So somebody can have malignant hyperthermia and they're totally normal walking around. They don't even know they have the disorder unless they've got a family history. They might have some suspicion. They go into a surgery, they're anesthetized, and the anesthesiologist will notice that the temperature is starting to creep up. The CO2 that they're exhaling starts to go up. And they start to get stiff muscles. What happens is these anesthetics, or some of the depolarizing muscle relaxants that they give you so that you don't get muscle contractions while they're doing a surgery on you, bind to the renin receptor, cause the channel to open. It dumps massive amounts of calcium into the cytoplasm, and basically all your muscles contract. It's like doing intense exercise. Metabolic rate goes way up. You get metabolic acidosis. You get nasty things. You get hypercapnia, cyanosis, heart rate goes up, muscle rigidity, high temperature, muscle damage. If the temperature, hyperthermia doesn't kill you, you get muscle damage, you link potassium into the blood. That can cause arrhythmias and cause cardiac arrest. Incidence, it's pretty rare. About 1 in 10,000 anesthetized children exhibit this, and about 1 in 50,000 adults. I was once talking to an anesthesiologist and asking why there might be such a difference. It's not that the incidence of the mutations are more common in kids, it's the way that they anesthetize children. If an adult goes in, they can explain to you what they're going to do. If a 2-year-old goes into surgery, it's kind of hard to explain what they're going to do. So they tend to administer anesthetics at a higher rate to children to get them to go under faster so they don't panic. And they think that's why the kids trigger more often than the adults. So about 70 to 80% of these cases of MH are linked to mutations in the renin receptor. There's a very, very large number of mutations. About 300 of them so far have been identified in the renin receptor. About 1% of the cases are linked to the L channel. And then there are a few other loci that have been identified, but the protein that's underlies the disease haven't been identified for those loci. So they've introduced dantrolene in the early 80s. And because of this, the incidence or the death rate from MH has dropped from about 80% to about 5% today. And most of these, this 5%, occur in outpatient surgery or these surgery centers that aren't in hospitals. So most hospitals today, most operating rooms, the anesthesiologists are very much aware of MH and similar disorders. and they have dantrolene on hand. So if you start to have any of these symptoms, they immediately stop the surgery, cool you down, and give you dantrolene. One other note, that folks who have this disorder, if you don't die on the operating table, because of this rhabdomyolysis, you leak a lot of proteins in the blood, and they can cause kidney failure in the days following. So just because you get through the initial crisis, they still have to keep a close eye on you. Also, there's been suggestions that MH will predispose people to heat stroke, to heat illness. Most of the time, this is triggered by these anesthetics. In some cases, there's some suggestion that extreme exercise and heat can also trigger it. In fact, in mice, a mouse model of MH, where they've knocked in the disorder, these mice are very susceptible to heat stress. You put them in a warm environment and they die off quite readily. Of course, these mice are generally homozygous, and most people are heterozygous, so they have a more extreme form of the disorder. But in those mice, this ICAR, which is, I don't know if you can see it because of the tables in the way, but ICAR is a AMP mimic, activates AMP kinase, but it also seems to have a prophylactic effect in this mouse model of MH. You give ICAR to these mice, and they survive the heat stress a lot better than the ones that haven't been given ICAR. ICAR seems to act through the adenine nucleotide binding site on the channel. Adenine nucleotides are well-known regulators of the channel. So folks who are working with ICAR seem to be targeting the adenine nucleotide binding site for therapeutic use. I don't have a slide summarizing central core disease, but it's also a rare disorder characterized by muscle weakness, and the central core is in histological sections of muscle fibers. You see that the core of the fibers has kind of this amorphous look to it, that there aren't contractile proteins and there aren't mitochondria there. So this lack of contractile proteins in the core of the fiber leads to the muscle weakness. And people with central core disease tend to have symptoms of malignant hyperthermia. The cardiac disorder, catecholaminergic polymorphic ventricular tachycardia. It's a autosomal dominant disorder of the Renin receptor. Also, there's a recessive form that's linked to a mutation in calsequestrin. Again, it's rare, about 1 in 10,000 people have it. And these folks are usually normal under sedentary conditions or at rest. But when they get stressed, particularly psychological or physical exertion, causes the release of catecholamines that cause irregularly timed calcium release from the SR. Calcium is released in the sarcoplasm, from the sarcoplasmic reticulum, goes up in the cytoplasm. The sodium-calcium exchanger is activated. It uses the electrochemical driving force for sodium entry to pump calcium out. The sodium entry causes depolarization of the surface of the membrane. You get an irregular, abnormally timed action potential, can lead to the arrhythmia. So this is one of the reasons for the sudden cardiac death in apparently healthy individuals. So you occasionally hear of athletes, high school athletes, who drop dead on the basketball court, and they have had no previous history of cardiac disorder. They do an autopsy, the heart looks fine, they can't tell what it is. They do a molecular diagnosis and they find mutations in the Renin receptor. There are lots of different causes of sudden cardiac death, but this is just one of them. So these symptoms appear around the ages of 6 to 10. You have these arrhythmias upon exercise or catecholamines. Syncope, people faint because of their arrhythmia. One of the first signs of the disorder is sudden death, and the mortality rate's about 30 to 35% by age of 30. The arrhythmogenic right ventricular dysplasia also predisposes people to arrhythmia, but there are clear signs of a disorder in the heart. You get fibrosis, fatty deposits, a lot of connective tissue in the right ventricle, so that can show up on medical exams. This doesn't. So, methods we use, two main ones: a radioligand binding assay using Ryanodine. So, Ryanodine binds to the ryanodine receptor, but it only binds when the channel's open. So if you radiolabel ryanodine, and it's commercially available, that's the structure of ryanodine. You mix ryanodine, radiolabeled ryanodine, with sarcoplasmic reticulum, and then you add something you think is going to either turn on or turn off the channel. You incubate, the ryanodine binds in the pore of the channel, basically gets stuck there. It has a very slow off rate. So after you do your incubation, you can filter away the excess ryanodine, leaving behind the receptor with the radiolabeled ryanodine stuck to it, put in your scintillation counter and count the radioactivity. If you have an increase in ryanodine binding, you know that you've opened the channel. If you have a decrease in ryanodine binding, you know you've closed the channel. The ryanodine binding is pretty quick, pretty simple. Single channel recording is a bit more tedious. In these experiments, you have a chamber that's divided in half. The divider has an aperture in it, a very small hole, maybe 100 to 200 microns in diameter. You fill both sides of the chamber with your recording solution. Then you actually paint lipid over this aperture. Initially, you have a little glob of lipid there. The lipid will spontaneously thin to a bilayer. You add channels, either sarcoplasmic reticulum vesicles or purified channels to one side of the bilayer, and you stir. Eventually, one of these channels will incorporate into the lipid bilayer. So you have a single channel in the bilayer. Ryanodine receptor, because it's kind of this mushroom shape, almost always inserts in the same direction so that the cytoplasmic face of the channel is facing the side where you added the channels. That way you know which side is cytoplasmic and which side is luminal of your channel. And you can add regulators to the cytoplasmic side if that's where you think the regulatory site is. Then you apply a voltage clamp across the membrane, and you control the membrane potential. So you can either record currents by having a gradient of a conducting ion, often cesium or potassium, sometimes calcium, or you can apply a membrane potential. You can have symmetrical solutions, apply a membrane potential, and drive ions through the channel when the channel opens. So when the channel is closed, you get basically a noisy baseline that looks something like this. So this is the closed state of the channel down here. So these are actual channel recordings. When the channel opens, ions flow through and you get a current. So you have this flickering back and forth of open and closed channels. So these channels are either open or closed. So if they're open, they have a characteristic conductance. You get a characteristic current for your conditions. If they're closed, you have no current. These small spikes here are usually due to limitations in your electronics. You just can't resolve very, very fast openings. So what we do with these experiments is first you note what percent of the time the channel's open. And you can also measure how long the channel opens when it's open or how long it stays closed when it's closed. And you can get an idea of the number of channel states. The channel goes from closed to open, but then there can be multiple closed and open states that have characteristic durations or distributions of openings and closings. So we've used these techniques to study this compound acetinosyl L-methionine, or SAM. So SAM is best known as a methyl group donor. You take SAM, which is present in your cells at about 100 micromolar or so. Then there's the enzymatic transfer of this methyl group to substrates, often proteins, nucleic acids, lipids, ending up with acetinosyl homocysteine. There's a paper back in about 2004, 2005 saying that ryanodine receptors from arterial smooth muscle were activated by this compound, and they suggested it worked through this methylation, so the enzymatic transfer of the methyl group. Smooth muscle is pretty heterogeneous. Lots of different, could have multiple different isoforms of ryanodine receptor in there. So we decided to try some of this stuff on our channel preps from either skeletal muscle or cardiac muscle. And what we saw is, yes, indeed, both cardiac and skeletal muscle SR were activated by SAM. So So you see this ryanodine binding. So if ryanodine binding goes up, you get, it means you've got more channels open, and then increasing concentration of SAM. So this steep part of the curve here corresponds to around the physiological concentration of SAM, but then it keeps increasing. Up to, this was 10 millimolar, was about as high as we could go. So we thought, hey, this is really neat. This would be a novel mechanism of regulating ryanodine receptors by methylation. Unfortunately, it's not the way it works. My student Angela Camphor did this work as part of her dissertation. And through a number of ways, including inhibiting methyltransferases or through radiolabeling ryanodine receptor with hot SAM, or incubating ryanodine receptors with hot SAM, where you would transfer radiolabeled the methyl group to whatever the target is. She would see that she got lots of radioactivity incorporated into sarcoplasmic reticulum, but when she immunoprecipitated the ryanodine receptor, she got very few counts of radioactivity. And then this was basically immunoprecipitation without the primary antibody. So if you immunoprecipitate the ryanodine receptor or you don't use the primary antibody, you get basically the same number of counts, indicating no, all this is background counts. So all this radioactivity is incorporated into something in the SR other than the ryanodine receptor. Ryanodine receptors only really make up a few percent of the protein in the SR, so the possible targets for this there's a huge number of targets. But like I said, this was kind of disappointing after quite a bit of work. So I said, well, let's go ahead and do some more experiments. Actually, that came after this experiment. This was looking at ryanodine binding, the concentration dependence of SAM or ATP, so this is SAM and ATP, and this AMP, PNP. This is a non-hydrolyzable analog of ATP. What you see is the SAM and ATP had the same concentration dependence. Here's the structure of ATP, here's the structure of SAM. Both SAM and ATP have this adenosine group. It's well known that adenine nucleotides bind the ryanodine receptor. So it seems that SAM was binding the adenine nucleotide binding site and activating the channel. This is a bit odd because ATP activates the channel. It's thought that the negative charges of these phosphates are required for channel activation, whereas SAM is actually positively charged, but it activated to the same extent as ATP. It was just a competition experiment where we do ryanodine binding in concentrations of ADP. This is the ADP activates. If you mix in ATP, you activate the channel. Increasing concentrations of ADP, you get no additive effect of the two, suggesting they activate via the same site. And the same result with SAM. You activate with SAM, increasing concentrations of ATP, there's no additive effect here, suggesting they activate the same site. So again, kind of disappointing because ATP was a well-known regulator. So I said, well, go ahead and do a couple channel experiments and we should get pretty much the same kinetic effects on the channel as from ATP and SAM. But we saw something kind of odd. So here is a ryanodine receptor that's been inserted into the bilayer, and in this case, we're holding the membrane potential at a negative potential. So in this case, channel openings, when the channel opens, current goes downward. When we add the SAM, what we saw was we get these subconductance states. I told you when channels open, they open to a characteristic conductance, and they close. They don't usually open halfway. So SAM was doing something funny, that you had these normal channel openings, but then you had these longer-lived subconductance states where the channel is opening part way. And even odder, I suppose, was at positive membrane potentials, at plus 50. Here, channel openings are upward, so this is closed, that's upward. These are openings here. When you added SAM, you actually inhibited the channel. You see long closings and very few openings. And this seemed to be dependent on this methyl group here, because when we did the same experiment with homocysteine, we didn't get the substates or the inhibition. Or if we use the methyltransferase inhibitor synfungin, which doesn't have the the sulfur in the methyl group, we also didn't get channel inhibition. This is just summary data. Here is the open probability of the channel. So this is the probability of finding the channel in the open state. And when you do these experiments, we set a threshold at 50% of the way between the open and closed state. Anything that exceeds 50% is considered open. Anything less than 50% is considered closed. Since this subconductance is about 2/3 the normal conductance, the subconductance is considered open. So that's why you get channel activation. That's at negative potentials. At positive potentials, where we get the inhibition, we have the concentration dependence of the inhibition. If you look at the ratio of the subconductance state normalized to the open probability, we see we get concentration dependence of the subconductance state also. So our question then became, well, okay, how does this stuff work? One of the first things we did was just took a closer look at the substates. So these are channel recordings to negative membrane potential, so openings are downward. These are on a slow time scale, multiple traces. Here's just one expanded. And here's with the SAM, we see the substates, and we see it on an expanded scale. If you look really closely here, you can see channels that go from the open state to the closed state. So if they're fully open, we assume they're not bound with SAM. They close and then they open to the substate, suggesting that the channel can go from the closed to the subconductance state. It doesn't have to go through the open state to get there. Or it can go from the open state to the subconductance state. So you have this triangular gating scheme where the channel goes from open to closed, which are these flickers here, and then to the substate, which are to here. Another interesting observation was that these substates, the prevalence of the substate was voltage dependent. At large negative membrane potentials, you had a relatively low probability of finding the substate. At less negative potentials, negative 25, you increase the probability of finding the substate, suggesting that the affinity of the channel for SAM seems to be voltage dependent. So ATP and SAM, well, we thought that SAM was acting via the adenine nucleotide site to turn on the channel. So was the substate also mediated via this ATP binding site? So to answer that question, what we do is we incorporated our channels, activated them with SAM, we got the substates, and then we added ATP, high concentrations of ATP, and what we saw was we had a reversal of the subconductance state, suggesting that ATP and SAM are competing for the same site. So binding ATP, you don't have the substate, you bind SAM, you have the substate. I should mention, these diagrams to the side are just amplitude histograms. So this is the current, amplitude of the current, and this is the probability. You can think of this as the number of openings. So you have lots of closings, which is zero conductance, and then you have some openings to a open level, and these are the subconductances. So there are some subconductances in here that just aren't too obvious. When you add the SAM, you see these subconductance, the probability of the subconductance state going way up. I just summary data, we see no SAM, we have a low probability of the substate. Add SAM, probability goes up, increase in concentration of ATP, the probability of the substate goes down. It is just the dwell times, how long the channel's open when it's open, how long it's closed when it's closed. Also, the inhibition at positive membrane potentials can also be at least partially reversed by ATP. So here's the channel and control conditions. With the SAM, you see the inhibition of fewer openings. You add ATP, and you get a lot of the openings back. So SAM seems to be working through the ATP binding site. But could it just be that when you add a channel activator, it overcomes the effects of the SAM? So we added this other channel activator, this 4-chloro-M-cresol, which is a pretty well characterized channel activator. What we see at plus 50, we have control traces with the openings going downward. 2 millimolar SAM causes a substate. You add the 4-CMC, and there's no reversal of the subconductance. At plus 50, openings are upward under control conditions. SAM inhibits the channel. You add the CMC, you get no reversal. So the reversal of these effects of SAM seem to be specific to ATP. So finally, we were trying to work out a gating scheme. So how does this SAM work? Well, we had the triangular scheme that we had already proposed, where the channel goes from closed to open, back and forth, and then goes to a substate. Well, if SAM, the duration of each of these states is going to be determined by the rate at which it exits the state. If SAM binds the open state and causes the substate with bound SAM, the ORN rate here will be concentration dependent. So the duration of this state, the open state, should be dependent on the concentration of SAM. Same with a closed state. If you can go from closed to the substate and SAM binds the closed state, this step should be concentration dependent. So if you measure the durations of the closed open state and duration of the substates, what we saw was that the duration of the open state didn't change with SAM concentration. The duration of the closed state didn't change, but the duration of the substate changed. So this suggests or tells us that the SAM wasn't binding the open and closed state of the channel, it was actually binding a substate. So if you went back, if you remember, I pointed out there was a small hump for in those amplitude histograms in the control state that suggests there was a substate. Now some of those were due to kinetic limitations of our system that we just couldn't resolve the very brief openings. But some of those were actually real. And it seemed to be that state that SAM was binding. So we ended up with this type of gating scheme. The channel goes from closed to open and then to the substate. We suggest that these transitions are voltage dependent. Remember that at large negative potentials, you had fewer substates than at small negative potentials. And this only applies to the negative membrane potential. SAM binds to the substate and stabilizes it. And then you can gate from the SAM substate to the closed state bound with SAM, because we saw that we could go from the closed to the substate or from the open to the substate. ATP competes with SAM by binding to the open or closed state. That was well known already, that ATP can bind to either state of the channel. And we'll convert the channel to a closed ATP bound state or an open ATP bound state, and actually it stabilizes it in the open conformation. So if you're up here with the ATP bound, you're less likely to enter the substate for SAM to bind. So that's the way we think that SAM is working. So right now we're working on how a gating scheme for the inhibition of the channel. So what we're interested in now is the channel subconductance and inhibition are mediated by the nucleotide binding site. And this could have implications for drug development, because as I mentioned, for malignant hyperthermia, there's that drug ICAR. It seems to bind the ATP binding site. And the current hypothesis, or the current thought of how ATP works, is that it requires the negative charges on the phosphate. Well, SAM can activate the channel, but it's positively charged. It also induces these substates. So if you think that the only thing that's going to happen when you manipulate the ATP binding sites, you're going to either open the channel or close it, you're going to be missing the effects of these substates. And I don't think you want to develop drugs targeting the site that are going to do things like that. Also, it's not known what the molecular features of the compounds that activate or work via the ATP binding site are. As I mentioned, they think it's the negative charges on the phosphate, but all those experiments that came up with that hypothesis only used phosphate-containing compounds. So if you get rid of a phosphate, not only are you changing the charge, but you're changing the bulk of the molecule. So we'd like to use a more diverse set of adenine-containing compounds, or adenosine-containing compounds, to see if we can tease apart the features of these molecules required for regulation of the channel. So I see I'm running kind of long here, so I'm going to be really brief on some of this other stuff. So we do some work with calmodulin. Calmodulin is a calcium binding protein. It's got 4 EF hands, 2 in an N terminal and 2 in the C terminal. It's kind of a compact conformation in a calcium-free or apocam state. When it binds calcium, it becomes more elongated, looks kind of like a dumbbell. In this form, exposes hydrophobic regions on both the N and C terminal that allow it to interact with proteins. So it activates or regulates ryanodine receptors in an isoform-specific manner. RYR1, again, we see ryanodine binding, so if you increase ryanodine binding, you increase the opening of the channel. And calcium concentration, so you get the bell-shaped calcium dependence in the absence of calmodulin. With calmodulin, you see increased binding at low calcium and decreased binding at high calcium. So calcium-free calmodulin seems to activate the channel. Calcium-bound calmodulin inhibits the channel. RYR2, you only get channel inhibition with calmodulin. For RYR1, this conversion from the activator to the inhibitor seems to be calcium binding to the calmodulin, because if you knock out the EF hands on calmodulin, you don't get channel inhibition. So here again, it's ryanodine binding, calcium concentration in your bell-shaped curve. Wild-type calmodulin kind of shifts the calcium dependence to lower calcium. You activate at lower calcium, inhibit at high. You knock out the EF hands, you only get activation. So calcium binding calmodulin causes inhibition. There are two models of how this works. It seems that calmodulin binds at least thought to bind two regions of the ryanodine receptor. One model says that calcium-free calmodulin activates the channel. Calcium binding the C-terminal EF hands causes inhibition. There's another model that says calcium-free calmodulin doesn't do anything, it just sits there. Calcium binding to the C-terminal EF hands causes activation. Calcium binding to the N-terminal causes the calmodulin to bind to the one peptide and to inhibit the channel. So what we did was went through and modified some of the calcium coordinating sites in the calmodulin to increase the binding affinity of individual EF hands. So these are the residues in calmodulin that coordinate the calciums. These filled-in residues are the ones that we modified. Then we measured intrinsic fluorescence of both the N and C terminal lobe. The N terminal lobe has phenylalanines that you can measure the intrinsic fluorescence. The C terminal has tyrosines, so you can distinguish between the C terminal and the N terminal calcium binding. And you can measure affinities of different mutants. Then we did our ryanodine binding, calcium dependence curves. The open symbols are no calmodulin. The open squares are the wild types. The filled symbols are different mutants for the N-terminal hands. If we mutate the N-terminal hands, we don't see any shift in the calcium concentration at which calmodulin goes from an activator to an inhibitor. So this calcium concentration where you convert from an activator to an inhibitor is we call it the calcium switch point. Should be related to the calcium affinity of the calmodulin. You increase the calcium affinity, you would expect calcium-bound calmodulin at lower calcium concentrations, and these switch points should move to the left. If you look at this on a different scale, so you have the calmodulin variant divided by the no-cam, you see that you get these sigmoidal curves, and if they shift to the left, it means if you increase you move the switch point to lower calciums. If it moves to the right, you move that switch point to higher calciums. So if we modify the C-terminal hands, we see some significant changes. And we see that the first EF hand, if we increase the calcium affinity of that EF hand, we inhibit the channel at lower calcium concentrations. If we modify the fourth EF hand, we shift the switch point to higher concentrations. This mutation, however, actually messed up the structure of calmodulin, so this had dramatic effects on the structure. This one maintained the structure of the calmodulin, was pretty specific to just modifying the calcium affinity. So in summary, if we modify the calcium affinity of the N-terminal hand with the C-terminal, we see that the one mutation in the N-terminal decreased the KD or increased the affinity for both. It decreased the calcium switch point, except this was not statistically significant, and we couldn't distinguish which EF hand was causing this. The C-terminal mutants were a little bit more informative. This third EF hand that we mutated decreased the affinity of the N-terminal, increased the affinity of the C-terminal, decreased the switch point. So the switch point seemed to be correlated more with the C-terminal affinity. The other mutant had the opposite effect, the fourth one, increase the affinity of the N-terminal, decrease the affinity of the C-terminal, increase the switch point. So this switch point seems to correlate with the change in the same direction as the C-terminal EF hands. So that would suggest this model is probably the more likely one, where we have apocalmogen activates the channel. Calcium binding to the C-terminal EF hands causes channel inhibition. That's just a part of the puzzle. We're doing more work with peptides from from the channel. We do know that the calmogen can bind either of these peptides, and we're working on which EF hand or which terminal binds which peptide. One thing we would like to do from this is go from the channel studies to muscle fibers. I mentioned that Ryanodine receptors can be activated by calcium, at least in a test tube, but in the muscle fiber, they're activated by this charge movement. So what we'd like to do is to do some experiments with muscle fibers where we can actually activate them through their normal physiological process. Where we actually take fibers, we can physically remove the surface membrane. When we do that, the transverse tubules actually will seal over. So here's a muscle fiber. You actually just grab a piece of the membrane with forceps and peel it down the membrane. The membrane actually kind of peels off like you're rolling down a sock. And what happens is as you tear the membrane here, here's the surface membrane, here's the transverse tube, and here's the SR. You tear the membrane, the T tubules seal, because you have this free membrane that doesn't like to associate with water, just associates with each other and traps the solution that you have the fiber in the transverse tubules. So if you're very careful in the way you manipulate your bathing solutions, you can polarize and depolarize the T tubules by changing the bathing solution. That will allow you to activate the muscle. If you hook your fiber to the single fiber that you've peeled to a force transducer, and you go from a high potassium solution to a high sodium solution, you cause those charge sensors, the L-type calcium channels, to move. You open the calcium release channels, release calcium, and you get muscle contraction, and you get force. The amount of force generated is going to be proportional to the amount of calcium released. You do that without calmodulin or with calmodulin, you see an increase in the amount of force generated, and you seem to see a faster contraction. So the reason you have to peel these fibers, it's very hard to get proteins in and out of muscle fibers. You can't use a patch clamp to do that because the diffusion distances are too long. Calmodulin binds to all the proteins in the fiber. And so this is one way you might better get calmodulin in and out of fibers. We also have calmodulin mutants, which are defective or have enhanced channel inhibition or channel activation. So we might be able to tease apart the role of calmodulin in voltage activation of muscle contraction, and use these mutants to determine the mechanism or the effects of calmodulin activation and calmodulin inhibition on muscle function. Lastly, and very quickly, just aging. As we get older, we get weaker. Part of it is due to alterations in calcium release and EC coupling. We've done some studies with old rat muscle and ryanodine binding to SR from the fast muscle or slow muscle. And what we see is old muscle has decreased activation by calcium, only from fast muscle but not from slow muscle. Calmodulin activation, so this is low calcium concentration. This should be apocalmogen. We see that the open symbols are the young muscle, filled ones are the old muscle. Apocalmogen activates both the EDL, or ryanodine receptors, from fast and slow muscle. The inhibition, though, is impaired. That this is calcium-bound calmodulin. There's no addition, increase in concentrations of calmodulin. We see inhibition of the ryanodine binding to the old channels or the young channels, but this reduction here with the old channels is not significant. But there is a reduction in ryanodine binding to SR from the slow muscle. So it seems that calmodulin and calcium regulation of old ryanodine receptors is impaired, but it only happens in fast muscle. It's kind of consistent with the observation that fast muscle is more affected by aging than slow muscle. I'll skip the ATP, there was no difference there. So lastly, what we'd like to do now is actually find out what the protein modifications of the renin receptor are that cause the impaired calcium regulation and CAM regulation. And finally, people involved with this, Angela Camphor, who is now Angela Gabordi since she got married, did almost all the same work. Priya Patel was a lab tech, and Andrew Schneider is another graduate student. I came from University of Minnesota where the calmodulin work started, so these guys, Brad and Razvan, were intimately involved with that. And then Georgia State, this group is made the calmodulin mutants and doing most of the intrinsic fluorescence and most of the protein biochemistry. I do most of the functional analysis. And folks at Marquette helped me with some of the peeled fiber work. And I've been funded by Heart Association and NIH. Sorry I went long. So, if there's any questions, and if you guys have to run off, I won't be offended.
So, I have a question. When you show the EDL and the soleus, is that the difference just in sensitivity, or is it just a difference in the anatomical or physiological grouping of these?
It's probably use, why there's an aging difference.
Well, not aging in the sense, but in terms of how the normal response works. Is there a difference between the fast and the slow?
No, you have the same isoform of all these proteins there. The amount of SR is greater in fast muscle, so you have more renin receptors per gram of muscle. But the isoform's there, so the sensitivity should be the same.
What I was thinking of was in terms of modulation. Do you think that there is a sensitivity modulator element somewhere along on the intracellular domain at least?
It would be more likely that it's some signaling pathway that impinges, that's present in slow or fast, some phosphorylation or reactive oxygen, some modification of the channel, and it wouldn't be intrinsic to the channel.
Thank you.
Okay.